Life teems with communication. From the intricate dances of bees to the haunting calls of whales, the natural world thrives on the exchange of information. But what about the communication within our own bodies? Cells, the fundamental building blocks of life, are in constant communication. They send and receive signals to regulate biological processes. This network ensures our bodies function in harmony. Cells send out signaling molecules like messengers carrying vital information. These messengers trigger specific responses. This process, known as cell signaling, is fundamental to life. In our cellular city, different types of communication occur. Some messages are whispered between neighbors. Autocrine signaling is like leaving yourself a note. This is crucial for processes like cell growth and differentiation. Paracrine signaling is akin to talking to your neighbors. This is important for coordinating local activities such as wound healing. Both rely on diffusion, moving molecules from high to low concentration. For long-distance communication, cells employ specialized messengers and dedicated pathways. Endocrine signaling is like sending a letter through the postal service. Hormones, the body's long-distance messengers, are secreted by endocrine glands into the bloodstream. They travel throughout the body, targeting cells with specific receptors. This allows for coordinated responses across different organs and tissues. Think of the hormone insulin produced by the pancreas. It regulates blood sugar levels by signaling to cells throughout the body to absorb glucose. Synaptic signaling is like a phone call, a rapid and precise form of communication. It occurs between nerve cells or neurons at specialized junctions called synapses. When an electrical signal reaches the end of a neuron, it triggers the release of neurotransmitters. These chemical messengers diffuse across the synapse and bind to receptors on the adjacent neuron, transmitting the signal. This intricate dance of electrical and chemical signaling allows for the rapid processing of information in our brains. Once a signal reaches its target cell, it embarks on a journey into the cell's interior. This is where intracellular signaling takes center stage, a complex relay system that transmits the message to the appropriate department within the cell. The cell's interior is a bustling metropolis of molecules, each with a specific role to play. The signal, often a small molecule called a ligand, binds to a receptor protein on the cell surface. This binding event triggers a cascade of intracellular events like a chain reaction. Think of it as a game of molecular tag. The ligand is it, and it tags the receptor, which then tags another molecule, and so on. This relay system amplifies the signal and ensures its precise delivery. Section 5. Messengers of the microscopic world. Secondary messengers. In this intricate dance of intracellular signaling, secondary messengers play a crucial role. These small molecules act as intermediaries, relaying the signal from the cell membrane to downstream targets. One well-known secondary messenger is cyclic AMP or CMP. When a ligand binds to certain types of receptors, it activates an enzyme called adenylyl cyclase. This enzyme, in turn, produces CMP from ATP, the cell's energy currency. CMP then activates other enzymes, like protein kinase A, which phosphorylate target proteins. This phosphorylation, the addition of a phosphate group, acts as a molecular switch, turning proteins on or off. Another important secondary messenger is calcium ions, or Ca2+. These ions are normally stored within specialized compartments inside the cell, such as the endoplasmic reticulum. When a signal activates certain receptors, it triggers the release of Ca2+ into the cytoplasm. This surge in calcium levels activates a variety of calcium-binding proteins, which then regulate various cellular processes, including muscle contraction, neurotransmitter release, and gene expression. Section 6. Gatekeepers of the cell, Ligand-Gated Ion Channels. The cell membrane, a selectively permeable barrier, controls what enters and exits the cell. Embedded within this membrane are specialized proteins called receptors, the gatekeepers of cellular communication. These receptors bind to specific signaling molecules, triggering a cellular response. Ligand-gated ion channels are like guarded doorways, controlling the flow of ions across the cell membrane. When a specific ligand binds to the receptor, it causes a conformational change, opening the channel and allowing ions to flow through. This flow of ions alters the electrical potential across the membrane, triggering a cellular response. One example is the nicotinic acetylcholine receptor found in muscle cells. When the neurotransmitter acetylcholine binds to this receptor, it opens a channel that allows sodium ions to flow into the cell. This influx of sodium ions depolarizes the muscle cell, triggering muscle contraction. These receptors are crucial for rapid signaling events such as nerve impulse transmission and muscle contraction. Section 7. The versatile communicators. G-protein coupled receptors. G-protein coupled receptors, or GPCRs, are the most numerous class of receptors in the human genome. They are involved in a wide range of physiological processes, from vision and smell to hormone response and neurotransmission. These receptors are like versatile messengers, relaying signals from a diverse array of ligands to a family of intracellular signaling molecules called G-proteins. When a ligand binds to a GPCR, it activates the associated G protein, which then interacts with other effector proteins, such as enzymes or ion channels. This activation cascade amplifies the signal and triggers a variety of cellular responses. One well-known example is the V-adrenergic receptor, which binds to the hormone adrenaline. Activation of this receptor triggers a cascade of events that ultimately lead to increased heart rate and blood pressure, preparing the body for a fight-or-flight response. GPCRs are involved in a vast array of physiological processes, making them important targets for drug development. Section 8. Growth and Development Architects, Tyrosine Kinase Receptors. Tyrosine kinase receptors, or RTKs, play a pivotal role in regulating cell growth, differentiation, and survival. These receptors are like construction foremen, receiving signals that instruct the cell to grow, divide, or specialize. RTKs are single-pass transmembrane proteins with an extracellular ligand-binding domain and an intracellular catalytic domain. Binding of a ligand to the extracellular domain activates the intracellular kinase domain, which then phosphorylates tyrosine residues on target proteins. This phosphorylation event acts as a molecular switch, activating downstream signaling pathways that control cell proliferation, differentiation, and survival. One important example is the epidermal growth factor receptor, or EGFR, which is overactivated in many types of cancer. Drugs that inhibit EGFR signaling have shown promise in treating certain types of cancer. RTKs are essential for normal development and tissue homeostasis, but their dysregulation can contribute to diseases like cancer. Section 9. Masters of Gene Expression, Nuclear Receptors. Deep within the cell, in the nucleus, lies the cell's genetic blueprint, DNA. Nuclear receptors, a unique class of receptors, act as the gatekeepers of gene expression, controlling which genes are turned on or off. These receptors are like transcription factors, binding to specific DNA sequences and regulating the transcription of nearby genes. Unlike other receptor types located on the cell membrane, nuclear receptors reside primarily in the cytoplasm or nucleus. When a ligand, often a hydrophobic molecule like a steroid hormone, binds to a nuclear receptor, it translocates to the nucleus and binds to specific DNA sequences called hormone response elements, or HREs. This binding event either activates or represses the transcription of nearby genes, ultimately affecting protein synthesis and cellular function. One example is the estrogen receptor, which is activated by the hormone estrogen. Binding of estrogen to its receptor regulates the expression of genes involved in female reproductive development and function. Nuclear receptors play a crucial role in a wide range of physiological processes, including metabolism, development, and reproduction. Section 10. Drugs and the art of cellular conversation. Drugs alter the body's function by interacting with cellular signaling pathways. Understanding drug-receptor interaction is crucial for developing therapies. Drugs can act as agonists or antagonists of receptors. Agonists mimic natural ligands, activating receptors and triggering responses. Antagonists block natural ligands, preventing receptor activation. Drug effectiveness is determined by potency and efficacy. Potency is the concentration needed for an effect. Efficacy is the maximum effect a drug can produce. Section 11. The Symphony of Signals. A final note. From the simplest single-celled organism to the most complex multicellular animal, cell signaling is the language of life. It is a symphony of signals, a constant conversation that ensures the harmonious functioning of every cell in our bodies. We have only just begun to decipher the complexities of this intricate communication network. Further exploration into the world of cell signaling holds immense promise for understanding the fundamental mechanisms of life and for developing new and effective treatments for a wide range of diseases.